Hello, thank you for listening in on my research project. My name is Madeline Tillman, and today I'm going to share with you the work I've been doing this year. My poster title is Evaluating the Chemical Complexity of Botanicals Active Against Drug-Resistant Bacteria. Dr. Chuck's lab is mostly focused on finding new natural product sources for the treatment of drug-resistant bacteria. My project involves Methicillin-resistant Staphylococcus aureus. For the remainder of this presentation, I refer to it as MRSA. Finding new treatments for MRSA has become more and more complicated due to the bacteria's high resistance to antibiotics. Our lab focuses on finding natural product sources to combat MRSA due to the diversity in chemical structures of molecules made by these natural product sources. Currently, 70% of small molecule treatments for infection, including well-known antibiotics like penicillin, are based on natural products. The selected botanicals of interest of this research are Stachys officinalis, Stachys peninsula, Hypericium calycium, and Ligustrum virgae. The goal of this project is to use mass spectrometry-based metabolomics to map compound distribution in the roots and leaves of each botanical, thus evaluating existing herbal remedies for compounds or groups of compounds which may contribute to the biological activity against MRSA. Moving down to my project workflow, there are three main steps that I took for this project. The first being the extraction of my botanicals. After all the extractions were finished, I tested the extracts against 2 strains of Staphylococcus aureus to assess their biological activity. The final step of this project is a metabolomic study where I will analyze the compound distribution in the extract to evaluate their chemical similarities and differences. Now I will walk you through the bioactivity screening of my samples. I ran 2 experiments using 2 strains of Staphylococcus aureus. The top graph shows data from the wild-type Staphylococcus aureus strain, whereas the bottom graph shows data from our MRSA strain. The main difference between these 2 strains is the strength of the bacteria. Wild-type Staphylococcus aureus is weaker than MRSA. On the x-axis of this graph is the botanical extracts. I've grouped these by plants for a clearer picture. On the y-axis shows the percent inhibition, or how much the bacteria was inhibited. The last bar is my positive control, levofloxacin, so we are looking for tall bars like this one. As you can see, for both Staphylococcus aureus strains, St. John's wort showed the most activity. In the metabolomics data processing methods, I am outlining 4 data processing steps of the metabolomics portion of this project. Step 1 is completed using MZMine to remove noise and deconvolute the features. As you can see, removal of the noise takes features that appear as real and removes them from the data set. Here we are going down from 4 features to 1. Deconvolution of features involves separating out features that collate together. In this example, the feature at the top has multiple peaks in it. Deconvoluting separates these out. We use Aureus to analyze the data further. In this step, a dendrogram and principal component analysis, or PCA plots, was generated. The dendrogram shows the relationship between the triplicate sample injections. The leaves, supplements, and roots appear to group together respectively, showing the data processing parameters are ideal. The PCA scores plot condenses the samples as an average of the triplicates. The distribution of the points on this plot shows that there is significant differences in the chemical profiles of the samples. The PCA loadings plot shows the features that are causing the differences in the scores plot. The last step I'm showing here is using Excalibur. This analyzes features from the PCA plots to determine compound identity and map distribution of features. Here I am showing the example from the loadings plot. I'm looking at the green dot. The selected ion chromatogram shows that this feature was detected in the leaves and the roots, but not the supplement. Further analysis is needed to determine the significance of this feature. Moving on to the conclusions, the goal of this project was to biologically assess the activity of my selected botanicals. Figure 1 shows the activity that was evaluated in 2 strains of S. aureus. In the less virulent wild-type S. aureus strain, St. John's wort roots and leaves were the most inhibitory of the bacterial growth. In the MRSA strain, St. John's wort also contributed to the most inhibition of bacterial growth. The drop in inhibition is likely due to the increased strength exhibited by MRSA in comparison to the wild type S. aureus. Future directions of this project include identifying the compounds responsible for the activity shown in Figure 1, as well as comparing the similarities and differences between compounds in the botanicals. I would like to take a moment to thank everyone in the Czech lab for providing guidance and training going towards the progress of this project. The Undergraduate Research Scholarship and Creativity Office supported this research project through the Undergraduate Research and Creativity Award and awarded the ERSCO Travel Fund to support travel and presentation of this project. I also want to take a moment to thank you all for joining in and listening.